I was testing our microphone, and now microphone would be working now. Three of the local participants, Dr. Murad and Dr. Asmar, Dr. Gil, please turn on your video, and then I just use the same scheme as the previous in September, that I'm going to ask to emphasize the topic and issue. Then I'm going to ask the local participants first by just naming, calling the names. The international participants, as you have already get very familiar with the sequential lectures of Professor Edmund Kim, you can just kind of anytime you unmute your instrument equipment, and then just to say your opinion or your answer. If the answers from the local participants, Dr. Murad and Dr. the Asmar Sarkar and Dr. Gil, Junyoung Gil's one was not so satisfactory to you, all of you, any of you. So, Dr. Murad, would you please turn on your video? Okay. So local participants are requested to turn on the video. Why? Because I'm going to use the interactive one. In September, it was a dream, but after Professor Edmund Kim's lecture, you might be become very much familiar with this, even more familiar than me. Okay, and then the Professor Dr. Hendra. We are now all prepared, and what we are waiting for is the hospitals beginning to give us the visa. So far, according to the social distancing scheme, they are not allowed to help visa qualification. So if visa is okay, then everything is prepared. Okay, so that's small information. And then before everyone just to join, let me just kind of make a little bit of diversion. Next week, you are going to listen to another lecture from Professor Ha, a young junior professor. However, who had been a 5-year or 6-year, 6 year after he received the boardman here in this country, and then after receiving 2 years or 3 years after receiving PhD. So in my opinion, he's one of the best faculty who can just, why? Because recently he get to know what can be done with the brain imaging. So that's next week. And after next week, without any kind of closing the announcement, it is going to this semester's the lecture series is going to be completed. So I wish you have enjoyed, and I'm quite sure that you would have enjoyed at least very much standard, and also the traditional lecture from Professor Kim, which is highly respected. Okay, and then let me just kind of release a little bit of our plan for next semester and the next year's second semester. At first, the professor, Dr. Hendra, gave me the feedback that the young doctors from Indonesia, they would like to listen to the correlative lectures regarding correlative imaging. So I asked Professor Im Professor Im is now the tenured, not still tenured, the official professor in Seoul National University in Graduate School of Science and Technology, Convergence of Science and Technology. So he's mainly involved in research now, but his lecture skill and also his expertise in correlative imaging was already confirmed by our Bangladesh colleagues. When I first visited Dhaka 2014, I coordinated a remote lecture. At the time, it was not so smooth, but we could. And then I had received the feedback who might have been the most helpful to them. And Professor Im, at that time the postdoc research scientist in my department as an alternative service to military one, and his lecture was regarded as the best, better than mine, okay, and other professors. And then he has published with Professor Edmund Kim, from Springer, SPECT, PET, and MRI. No, SPECT and PET, SPECT CT and PET CT, and PET MRI. Well, that book was almost handled by Professor Im. So Professor Im and Professor Soo, both of them, I asked them to take care of the planning, the lecture series regarding the correlative imaging between the anatomy and our nuclear medicine imaging. So I think we can start in the middle of the February, not just waiting for March, and weekly, the same Wednesday. And then why we start a little bit early? It's because of the Children's Day is Wednesday once, and one holiday, and another one is Buddhist Day is also Wednesday. So next year, spring semester, we would lose two Wednesdays. So I am planning to start our lecture in the middle of February, and then it is going to continue almost to 10 to 12 lectures, 10 to 12 and then yeah. So I'm still allowing the entrance. Dr. Otomo is just here, just arrived. So and then Dr. Im and Dr. Soo's lecture, and they are not going to be the only persons, and they are going to invite best people from my country, and from Jeonnam National University or the Gyeongbuk National University or other hospitals such as the Samsung, if they agree, Samsung or the Asan Medical Center, Severance, and or even Borame Hospital or National Cancer Center. All those papers are going to be consulted now, and then those kinds of lectures are going to be continue. I mean, not continue. Just we are going to plan the 10 lectures maybe, and then 5 for a spring semester and 5 for a fall semester, and then I have successfully invited Professor Pang. He recently formulated a good lecture about the basics of myocardial SPECT and other nuclear cardiology. So he's going to be responsible for 3 consecutive lectures, including the Professor Cho. Professor Cho is the kind of a follower of Professor Peng in terms of the nuclear cardiology in this country. And also I successfully invited the Professor Choi Hong-yoon. Choi Hong-yoon would have been the resident at that time, but he's after serving military service, coming back from the military service, he just immediately had become the clinical professor. He's now in charge of the departmental administrator. So he can just tell you about the cutting edge topics of the genomics. No, not genomics. The multi-omics, which can be associated, can be combined with the nuclear medicine imaging and to be used for improving the patient care. So you are more than entitled. Why? Because you belong to the future. As the next generation leaders of the Asian country, just like RCEP has not included the regional one. Just all the prime ministers and the presidents had signed recently. 15 countries along from Japan to Myanmar, and it did not include the Pakistan, India, and Nepal, Bhutan, and Bangladesh right now. Anyway, most of the Asian people are under free trade, while Afroza. Okay, finally, I think everyone has just arrived. So let me start my lecture. Okay, so you can just kind of interrupt me anytime. And I have sufficient and scanty amount of slides, and I have two topics here. In my opinion, if we do not explore the possibility of theranostics or therapy, then the nuclear medicine entire field is doomed. One reason is the introduction of deep learning or artificial intelligence. Imaging the specialty or expertise is going to be surrender to the use of the computer, and then the future generation, future humankind would not need so many radiologists or nuclear medicine physicians who majored in image reading. Image reading is just kind of a skill. So we need to just develop therapy or theranostics. I'm going to explain molecular theranostics, not a usual way. So you would have already browsed my the topics, and some of them are in handwritten one. So you would not find my contents for the first half of my lecture today, my presentation today. You would not be able to find the contents in the textbooks. If they are in the textbooks, there is no need to just explain. Okay, then everyone can just read a textbook or review article or the. Okay, still Dr. Prima. All right. So I have just kind of procrastinated or just prolonged my introduction so that everyone can enjoy the real contents. And today, so I had just kind of put the side or the kind of a sub title here: solutions and problems to solve. Those problems are not going to be the best people. What I'm saying, the best institution and then runner-up what I'm saying is just pioneers. Pioneers do not care the real problems to be applied for the clinical situation and following institution and following generation and following countries. We have experienced, I myself have experienced that kind of situation 3 decades ago. And it was a really, really embarrassing and at a loss and unsatisfactory and uncomfortable situation. We could not understand why the leaders and pioneers in the Western countries is interested in some problems, and sometimes it looked like they are interested in trivial ones and peripheral ones, or maybe the cutting edge, and they are seeking for just kind of a rarity or the peculiarity, the common. They do not say anything about the common, and they do just once we invite one of them, and they do just can teach us only the one aspect of the entire nuclear medicine. So the 40 years ago and 30 years ago, we invited Professor Edmund Kim and several times, and he gave us I remember the time that I just was one of the audience in his lecture regarding renal scintigraphy at the time. Until listening to his lecture, after reading so many textbooks and review articles and JNM or European Journal articles, I could not understand anything. So they were just kind of a waste and garbage. But after listening to Professor Edmund Kim, and he was so simple and easy to understand. And I was surprised that he's not a nephrologist, and he's just kind of a radiologist turned into nuclear medicine physician. So I was surprised, and from that time I respected him, and that's the reason why I'm just kind of I have planned the distinguished lecture series by him, and I really appreciate his the input. Anyway, following his one, I'm going to be a very easy to understand. But if you don't understand, then I'm going to explain in the next semester and the next to the next semester, whatever. And the second one is I have given the lecture in Vietnam and Bangladesh, and also the Indonesia. So several countries regarding radionanomedicine, and also WORMS meaning and World Association of Radiopharmaceutical and Molecular Therapy. So that WORM meeting, last year's WORM meeting was in Nanjing, and I have given in brain the radiotheranostics, and it also includes radionanomedicine. And also today, this year's one, I have recorded with Professor Soo. Then, and in December, you can just, if you have any chance to listen to our presentation, it's 20 minutes long, and we'll explain about the future of nuclear medicine. This is not actually the entire future of nuclear medicine, and this is just kind of a small part of the future, which is expanding from small molecules to some larger ones, which is called nano particles or nanomaterials. And I have devoted to learn and understand and apply and investigate the possibility of using nanotechnology in nuclear medicine for now 11 years. Obviously, I certainly remember when I started. It was just 2009 February when I took a tour, took a travel to Postec. It's kind of a well-respected the engineering college, science and engineering college in the southeastern part of my country, and we visited. We meaning I and my colleague scientists. We visited several of the nanotechnology experts who had just came from the United States and Europe, and to become the faculty, and we discussed and learned many things. Okay, so you might just kind of expect. I think I have used enough time for introduction, 20 minutes. Then for the remaining 40 minutes, I'm going to explain the molecular theranostics and core concept of this one. As I just told you, those contents are not going to be found in the textbook. I have derived some of them from a review article, but the interpretation is mine. Okay, what remains to be solved? You are entitled to listen to this one. Why? Because nothing has been solved. So still, this is going to be this is already being used, and the big pharma had merged and acquired with a huge amount of money from AAA, Endosite, or whatever other the companies, the startup companies. It does not mean anything. So what remains to be solved, I'm going to explain. So problems to be understood is the first one is why alpha therapy, actinium and lutetium for therapy, differential characteristics. After explaining that one, the main part of this first half is just external beam radiation. External beam radiation has been established for such a long decades, several decades, and which is now essential for every country. If you are interested in treat and cure the combined chemotherapy and radiotherapy is a kind of a standard. What about nuclear medicine therapy? It's kind of exotic and peripheral, and not so the well publicized one. So based on the knowledge which was already available, which is already available, external beam radiation and biological effective dose or something, and I'm going to explain the radionuclide molecular therapy. And then for a short while, effectiveness evaluation of new therapeutics, and then dosimetry, especially individual for maximizing effect and efficacy. That's the problem you need to know. And then safety issues and new direction. And for new direction, I'm going to explain among many new directions, explain radionanomedicine as I just told you. At first, actinium and lutetium for therapy. We need to just kind of familiarize ourselves in the location of these materials. Actinium, before actinium is just atomic number, one less is radium. And also the one high is thorium. And here, and let us just kind of get familiarized ourselves. And lutetium. Lutetium is here. You know, the first row is lanthanide. The first one is lanthanum. And then second one was actinium. So it's lanthanide and actinide. I'm not quite sure how it is called in English. Anyway, the lanthanum and then actinium is the kind of leading the atom, and lutetium is located here. Before it ytterbium, and after it is hafnium. So please don't bother radium, don't bother thorium or ytterbium or hafnium. What you need to know is just kind of the familiar one. The upper row, there is an yttrium. So that's the reason why yttrium and lutetium is always coming together. Lr might be Laurentian, I forgot. And here, lanthanum, actinium, and UT is just unknown. Okay, for some reason, the PRRT has become very familiar. And in my the understanding, current PRRT is only consists of current PRRT consists only of two. One is the SST2 ligand, which is somatostatin receptor 2, and octreotide from Sandoz, previously Sandoz, and which is called sandostatin, and which was developed to suppress the pituitary adenoma at the time. 30 years ago, I used sandostatin to test the FSH producing pituitary adenoma. And when I injected sandostatin, the FSH was just decreased. The long story short, and DOTA talk and DOTA tape. In my country, not the yttrium here, the gadolinium labeled DOTA talk is used for imaging. Why? Because for some the unknown reason, Korean FDA approved it first, not from DOTA tape. And then second one is folate hydrolase 1. Actually, PSMA has many names. And the Martin Pomp of Johns Hopkins and his colleague Steve Joe in Wisconsin, Steve Joe explained the pioneering days of Martin Pomper and Johns Hopkins. Martin Pomper has just to recognize that the folate hydrolase and some kind of a decarboxylase, glutamyl or something, and PSMA are all the same the enzyme on the surface of the prostate. And interestingly, like thyroid cancer cells, prostate glandular cells are going to be mad, crazy to become the prostate cancer. However, they do not lose. Instead, they overexpress the folate hydrolase. And folate hydrolase is recognizing this compound here. Here, glutamate urea lysine. Usually we use the one syllable for amino acid. However, in this case, because of the middle location of the urea, they have already used the GUL. We use the K for lysine. However, in this case, GUL. So they all say that GUL. Here you can see the prostate-specific membrane antigen has a pouch to recognize the urea and then glutamated lysine. So the third compound, lysine, should be always lysine? No. It can be glutamine, it can be others, ornithine and whatever. However, the GUL has in some way had become the first to be applied, and once it's kind of a pathway dependent. There has been no bias to use the GUL instead of the GUGN, the glutamate urea glutamine or glutamate urea other compounds, other amino acid. However, so PRRT is based upon the success of actually the unique characteristics of the tumor. Neuroendocrine tumor has a very high the propensity on their surface and somatostatin 2, and then prostate cancer has a folate hydrolase 1. Okay, so we are using this tool. The lutetium has a beta emitting beta ray here. Higher is better or lower is better. And recently we started to use the actinium, and we are waiting for the astatine. And radium is already used for treating bone metastasis by the pioneering works of the Norwegian the nuclear medicine physicians in, as I remember, Radium Institute or something. And their half-life is a little bit different. However, in case of astatine, we need to understand the decay diagram. Anyway, let me just let us memorize that the alpha particle has a range of the penetration of 0.1 millimeter, and then beta particles has a variety of energy here. So radium is already, as I just told you, already used. Radium-223. The commercial name is Zofigo. The 20 years ago, 30 years ago, we tried to use the strontium or samarium. That was my mission. I have been given from my the late Professor Ko to try to use. And after looking into the literature, I found that the strontium and samarium does not help the people to survive, or the progression-free survive, or the quality of life. So I just reported to Professor Ko. I don't think that strontium and samarium, which is so expensive, is going to help our people. So we stopped. So according to the Dr. Yoshida, Japanese journal here, single sentence is very important, very easy to understand. Compared with strontium-89, radium-223 has a shorter physical half-life, which is a benefit. Quite not sure, but this is important. Shorter tissue range, 0.1, and the most important one is higher linear energy transfer. So prostate cancer, if they have metastasized and sitting down and just kind of stimulating osteoblastic activity, which is giving the bone scan of a disseminated metastasis, is going to be killed by the radium. Why? Because the radium chloride is a bone seeker, like a bone scan agent. And then what about the lutetium and yttrium? Which one to choose and why? There was the real problem when the Steve Jobs of Apple company had found that he had developed a neuroendocrine tumor. And then using his he's so wealthy and he's a celebrity, so he might have reached every institutions. But interestingly, he did not choose Richard Baum's Bad Berka. He chose Basel. And at that time, the which one to choose between lutetium and yttrium was not well established. And also, according to his biography, that strangely, Steve Jobs was a advocate of comprehensive and the alternative medicine. So he lost the golden time. And then at that time, we did not know which one, lutetium and yttrium. Then now, after almost 10 or more years, do we know which one to use, the lutetium or yttrium? I don't think so. So I can say that this is kind of no man's land, and it's kind of uncharted territory, and you need to step in. And to step in that uncharted territory, you need to understand this. Yttrium has a 2.3 mega electron volt, and lutetium has a 0.5 mega electron volt, meaning lutetium is a low energy beta, and yttrium is a high energy beta. And if you are going to inject the same amount, people might think that the high energy beta would be better than low energy beta. So interestingly, most of the nuclear medicine physicians do prefer use the lutetium-177. So we need to understand why. So is it because of the because of the range, meaning tissue penetration. However, in case of the radium and the strontium, as I just told you, tissue range is shorter for radium, but radium is effective. Then tissue range is not a problem, and 2.3 mega electron volt is more important. Then why we are using lutetium lutate instead of the yttrium dotatate? And we move on the actinium instead of the lutetium, from lutetium to it might be it might have been the availability, but from lutetium to yttrium, no, from lutetium to actinium. So after my just lecture today, at least you need to understand, or you need to find the clue for yourself on your own to find out why. So every big shot or pioneers in their lecture, they don't say anything about the why lutetium over yttrium. If he asked them, they just answer, you need to do the more clinical trials to find out which one would be better. So while doing waiting for the clinical trials, people are dying, and our discipline is decaying, and the gloomy or grim future prospects for the next generation of nuclear medicine. And there is another variable parameter here. Lutetium has a half-life of 7 days, and yttrium has a half-life of 3 days. Usually half-life is the short, then we can just inject the more amount of the radioactive substance, such as in case of thallium, we need to just inject less than 1 millicurie. In case of technetium MIBI, we inject 20 millicurie. So that's the situation. So by just looking at the parameter, I could just expect that yttrium might be better than lutetium, but then it's contradictory to the current trend. So this is the question that I'm asking. So then people can say that why don't we just combine lutetium and yttrium? Shorter range is going to be devoted to lutetium, and a longer range and a farther one. And why? Because any kind of the compound might not reach the target very well. Why? Because of the necrosis and apoptosis and poor vascular supply. So one can simply we don't do this now. However, for example, if you were to combine, then we need to just combine 2.3 mega electron volt and 0.5 and 3 days and 7 days. And then once you inject, and then 7 days, and the entire amount of radiation would be higher with the same amount of the here. And then so to simplify the parameter, we don't use the initial injected amount and the half-life of that one. And actually that half-life contains not just the physical half-life, it should include the biological half-life, and we call it effective half-life. And then anyway, that biological half-life is associated with the renal excretion or the intestinal excretion, or it's just kind of a blood circulation time and whatever. So we are going to use the residence time. So meaning that entire area is divided by the injected amount, and then which is going to give us this kind of concept. This is no, not this one. This is the residence time, and this and it's the same. So you can just easily understand. This is the elementary or the kindergarten type of information. Now, this is all. I just raised a question. I did not answer. Why? Because nobody knows. So you need to just determine. And to understand the second problem, problems to be understood. The first one is, the second one is the comparing the external beam radiation. Why? Because for external beam radiation, our radiation oncologist colleagues, they have promoted, they have just kind of developed a lot of, collected a lot of knowledge regarding how to put the external beam for radiation, successful radiation, to just to save people. So we are the kind of a latecomer, new kids on the block. So why not understand the external beam radiation? So we need to just understand the, but once you're interested in to find out, to purchase a radiation oncology textbook and to just to open the chapter for physics, it is, or the biology, radiation biology is beyond your understanding. There are so many unnecessary details are there. So let me just summarize it. The most important concept is biological effective dose here. You need to understand only this one. Okay. So for biological effective dose, in many articles, 2012, the Physics in Medicine Biology, and also the or JNM, mostly Physics in Medicine and Biology. Biological effective dose, and they try to just to explain the usually physicists or the radiation biologists or combined one. Best experts are describing this one in review articles, and many things are, and they are going to say that disease control probability, and they compare and then also this kind of new information is usually included, and we don't need that. What we need to is just kind of what is biological effective dose and how to use it for clinical purpose. So the kind of they would have explained the tumor control probability 90% or something. We need just to put 180 gray. Then all of a sudden we have used the millicurie, and then looking at the image, and we were so confident and satisfied with the image quality. But now we need to just kind of inject something. And then how much amount of gray? Once a patient asks us that how much amount of the radiation I'm receiving, or how much amount of my tumor is receiving, can we explain to them? It's not it's beyond the patient's understanding. Only because the patients do not need to know that. Please, put your hands on your heart. Is it so? Isn't it because you don't understand how much amount of radiation you are going to give to the patient's tumor? That's a fact. We are disguising that we understand everything. No. And we only understand the preclinical data, and that it should be translated to the clinical situation. So the earlier work by the Konienberg, the Marion de Jong, the pioneers in this field, and Marion de Jong is an MD, Konienberg is PhD. They have done, and Sweden and Netherlands and also the United States, those people had done all this kind of approach, but still we don't understand. We meaning the normal or usual or at the clinic, we don't understand even the concept of biological effective dose. So I was trying to explain this in English. Actually, I can explain this in Korean very well, but please allow me to use English. The first one is survival fraction. Survival fraction is here, is reminding us the in vitro situation. So how many cells are dead or not can be counted. And if survival fraction is 1, then everything is alive. 0, everything is dead. But interestingly, they are using minus log, the natural log of survival fraction, which is giving us this kind of curve. And they are doing this one, ln SF, reverse one. Why? Because we just get rid of minus. All right? And then we can just assume that those cancer cell or normal cell has intrinsic sensitivity to radiation sensitivity, meaning intrinsic tendency to die with it's a certain amount of radiation. So this can be like this. Any kind of final results, any kind of final results can have the starting point, and any kind of one, even you can just imagine this, but this is not observed. This one and this one has initial slope. Okay, initial slope can be considered as an intrinsic sensitivity. However, all the cells have the capability to repair the DNA breakage. DNA breakage is a single strand or double strand, which is belongs to the realm of radiation biology, but we don't care. Anyway, the tumor cells are alive or not. So our colleagues, the radiation oncologists, several decades ago, they have developed here linear accelerated aperture is rotating here, and then this area normal tissue is receiving only once. Tumor is receiving several times. So that's kind of a spatial fractionation. And after some while, they have found this kind of one. Initial dose, single dose might be 100 gray, might be fractionated here. If we just kind of use the 10 and the one time 10 gray, usually we fractionate 50 times. Why? Because one time, every time they are receiving only 2 gray. 2 gray for spatial fractionation. And then they expect the normal tissues have the higher repair rate, and tumor tissues, they wish tumor tissues do not have such a kind of a versatility, resilience and robustness, and then they just kind of give up. So that's their tactic strategy, and and that's kind of this one. This one is just kind of this one. Here, fractionation here. d over n, d over n, d over n, d over n. The last night I just draw on my own. What will happen? Finally, we observe that this kind of quadratic curve is just kind of the linear form and then quadratic form. So this is a quadratic curve, and if we put minus, then the curve looks like this one. So empirically, experimentally, they put here d and d square. And then alpha is a initial inherent sensitivity here. And then all the others are considered to be BED. That's it. So BED for EBRT is like this one. So we don't divide initial D, this one. Why? Because this is inherent. It's not related with the amount of the dose. So then this is kind of very easy just interpretation. That's mine. Actually, no textbook has given this kind of information. It's interpretation. So if you just kind of put alpha BED here, alpha BED. So BED is like this, and then the second one is just kind of here. So d over n is d, small d, and then alpha BED meaning alpha BED, alpha, and alpha is gone, and then the n beta is here, beta of d over n. So you can think it might be n beta over n, or beta is here and d is root n divided by root n, whatever. So that's kind of the equal interpretation, and why not choose one of them? So next one is like this. If you decrease the amount and then the beta, this is here. Here, SF survival fraction 0 is good. 1 is bad. We do the dose, but not dose of n. So this is kind of, so beta is the explaining this. But if you divide d over n, then n is going to modify beta, meaning more cancer cells are surviving. Okay. So we need to just give again and again, and I just divide in 2 again and again, so that SF0 survival fraction 0 is achieved. Now you understand let me just assume that you understand the core concept of the linear quadratic model here, and survival fraction, and natural log. You might have you might have forgotten everything, this kind of elementary mathematics now, but that mathematics is used for the promoting your understanding. Okay. Then I will just emphasize here. Radiation is very good one. Why? It's because radiation is cytotoxic. Tyrosine kinase inhibitor or siRNA, they are all cytostatic. So it is not going to cure. It is just kind of a progression free. but we want to help people by saving them, not just kind of. Now, at this third slide, I have drawn the same. Now you understand the meaning of this alpha d plus beta d square. Sometimes alpha over d is used for the proliferation rate of the cancer cells or normal cells, and you might just use this one. This is an introduction of a huge concept which is well established and used for several decades to have saved a lot of people globally, and then kind of a core concept which is trusted by the radiation oncologist colleagues. But we are entering into the field, discipline of radiation oncology by doing the therapeutics or therapy or the theranostics. Okay. Then what about the molecular radionuclide therapy? We call it MRT. Now you are quite familiar with this kind of the nomenclature here, equation. So you are familiar, and you are also familiar with this. But according to the literature, Cremonesi, here, next paper. Where is it? Cremonesi. Yeah, here. Cremonesi. Please read this review paper. This is really good. Okay. I have derived all my the interpretation from this one. This review paper, page 6, only page 6. That review paper is 20 pages long, but what we need is just one page. Okay. And here they have used this kind of. I found that I was just trying to let you know that d over n. Finally, I found that d over n is d over lambda. Maybe might be the decay constant of effective decay constant. Okay, so I was just last night I just draw this the equation, and in their explanation, this is also the same as this one. Okay, and then instead of beta over n, we need to understand beta over lambda effective. Why? Lambda effective, the multiplied by t half is natural log 2, 0.693. If lambda effective is low, then the t half is high, and then t half is high, meaning dose rate over time, dose is high. So total dose is injected dose multiplied by residence time. Okay, I explained just a while ago. I explained to you that this amount of radiation is the same as this amount of residence time and initial injected dose. That's kind of a way of thinking. So you can just find that injected dose multiplied by residence time, meaning injected dose divided by lambda effective. So you can say that lambda effective is the same as the n fractionation. So now everything is clear. Now you can just follow Cremonesi and other big shots in radiation physics, therapy physics, and dosimetry, and also radiation biology. There is no magic. It's just kind of a physics, and which can be easily done. Why? I think that you have chosen nuclear medicine because you do not abhor, I mean, you do not hate mathematics or you do not hate physics. Physics is just physics and mathematics is physics. Mathematics is just mathematics. What you need is just how to understand and how to help and how to answer which one to choose between yttrium and lutetium. That kind of issues are the best kind of objective of ours, not understanding physics. Physics is physics. Okay, so this is one. And then I just draw another to assure you again and again similarity and difference between EBRT and MRT. In terms of, I have made a radiobiological effect models. Here, lower dose and loss of effect, that kind of, if you just kind of infinitesimally small fractionation. Okay, so later on, you might just enjoy this kind of drawing to let you understand. Once you understand, please don't look back. You just move on. You have understood some. So here, but you know, lambda effective is associated with mu repair. Mu repair has the same term, fraction per minute or fraction per day or fraction per, that's repair rate. An effective decay rate. Those rates can be combined. How to combine? They have combined, our physicists, eminent physicist colleagues, they have already combined. Once you think about the cells can repair, normal cells can repair better, and the cancer cells repair poorer, then we have chance to kill only the cancer cells by fractionation, spatial fractionation, and temporal fractionation combination. So modifies effect here, lambda effective was divided by here, lambda effective plus mu, right? So you can just combine in any way. So the final conclusion was that. So I just kind of put if repair is zero, meaning if repair is zero, then this is just kind of lambda effective over lambda effective, meaning the n seems to be not there. So beta over lambda effective, meaning it 1, then beta is working. Then this is better. So tumor has a repair capability of really poorest one, then tumor cells are killed, not just this one. So meaning our molecular radiotherapy in principle is better than the EBRT fractionated one. EBRT people, if they disregard the tissue penetration, they want to use the as small amount of radiation as possible. But we already use that. Why? Because we are injecting them and to let the radionuclide and radiopharmaceutical to be collected at the target site. So later on, please, you just kind of consider on your own. Okay. All right. Then finally, I just summarize this this one here with the real data. Interestingly, they have already they measured using the in vitro experiment. The proliferation rate might be per hour 0.28, 0.53. On yttrium, in this case, the Usually lambda effective one is 0.01 per hour and real values. So if T effective half is something, then one might be the results. So we can just calculate this one here, Cremonese. So I have reviewed many review articles and many the textbooks, and I found that this paper is most easiest one to understand or to develop your own understanding. Once you think you understand, you just kind of come back to this subject only when you are going to teach your juniors or students, medical students, to attract, to invite them to our discipline as a nuclear medicine physician, or more importantly, as a user of molecular radiotherapy. So, and I'm very much I'm really happy to have Richard Baum. His pioneering work has enabled us to have a good example. In that sense, thyroid cancer successful treatment is not a good example. In that case, everyone is treated. There is no need to understand the molecular radiotherapy in case of thyroid cancer. Thyroid cancer is the easiest one to treat, but we have so many cancers the posing problems and losing our patients' lives. So here, in this another the same paper, and they have explained this like this, and you might just kind of a EBRT and radioembolization and this one, and they needed to calculate this kind of parameter, equivalent uniform BED, meaning we have a liver here, cancer. If you just radioembolize, CERT here, selective internal radiation therapy, catheterization, and then yttrium, we need to do PET. Data image is so poor. However, we can do yttrium 90, or lutetium has a gamma ray, so SPECT. So then using SPECT here, some of them has a higher uptake, some of them not. But we need to treat entire cancer, not just a part of that. So if we just use the here and then use the histogram, how much amount of the lutetium has been, no, not lutetium, in this case, the usually yttrium. Yttrium has accumulated in, and then this is calculated equivalent uniform BED. BED is the mean, this one, that kind of calculation. So we don't need to be bothered. So they can use any kind of abbreviation, any kind of. We already understood the lambda effective and lambda the mu repair and alpha over beta. We are not intimidated any kind of equation. That's a very simple equation, and everybody can understand. The log, it's a log quadratic model. It's just a model. So thus, molecular radiation therapy, I'm finishing the first part here. This is my kind of take-home message. If T effective half is long, then linear response, negligible one. Linear response meaning it is good. And not good, linear response. And if T biological half-life is short, we need to increase the circulation time. If tumor retains therapeutic RI molecules much more than the remaining body does, we just can't care about the tumor only. So targeting is important. For imaging experts, nuclear medicine physician, I have spent almost 3 decades for image reading, mainly heart and brain. Actually, previously, when I started, liver and thyroid. And of course, bone. And WBC, whatever, and any other general nuclear medicine. And finally, I did heart and brain. When I just kind of entered into the field of brain, around that time, just 2010 or something, 2000 or something, until 2000, there had been rare FDG PET, no attenuation correction, and image was so poor. I was not interested in reading FDG PET, so I hated FDG PET, and after all, I found that PET-CT, not just PET-CT, PET-MRI is a good item for brain. So anyway, all of us are very good at targeting. And then if a tumor retains therapeutic RI molecules, remaining body is almost just like FAP. Most recent one, fibroblast-associated protein, is giving us or what? Not choline, but PSMA. Those images are so good. But in case of the FAP, everyone says that it is not targeting, just like a radium. Radium chloride is targeting osteoblastic cells, not tumors. Tumor is there, and fibroblasts are around there or the inside there. And FAP molecule is targeting, so we don't know lutetium FAP or the actinium FAP is working or not. So, and finally, difference between yttrium and lutetium. So the first one, again, 2.3 megaelectron volt, 0.5 megaelectron volt, and 10 millicurie and 5 millicurie, 3 days and 7 days. And this kind of one, lutetium has a lower energy, then intrinsic sensitivity is low. So this is good. Why? Survival fraction is here. Survival fraction is 1. Here, survival fraction is 0. This is good. Yttrium looks better, but still interesting. Even though, based on my explanation, even though the yttrium looks better, people are using lutetium. Why? So that's kind of Dr. Asmal, Dr. Gil. This is a term examination. You need to explain on your own way. According to my explanation, my interpretation of the molecular radiotherapy or even external beam radiotherapy, and the linear quadratic model or natural log of survival fraction or fractionation or whatever, still we expect the yttrium would be better. Then why people are using lutetium? That's the question. So I used almost 1 hour to explain my question in your term examination. So I used to release the question beforehand. You have 3 weeks to just devise your own answer. You can just kind of, so why? Because Nobody is answering that question. So you can just answer on your own way. You can be wrong in your own way. But what I'm proposing is you need to just think about that. You don't need to just we don't need to copy the everything presented or said by the pioneers or the world experts. They are so busy to do the next job, but we should be busy between the choose the options, choose between options such as the. Then let me just add here. Then alpha. So we have already radium here. We have already actinium here. We have astatine. Astatine is being produced by Fukushima. You know, the Fukushima has suffered from the tsunami and then explosion of the nuclear power plant and contamination. Many people were displaced. They have experienced, our Japanese colleagues, they have experienced misery. In Fukushima, they have installed a facility to produce astatine. Astatine is just a kind of fluoride, the iodine, and then astatine. Yeah? Fluoride, fluorine and bromine, and the iodine, and then astatine. Astatine is just kind of halide. And actinium and radium, I just let you know where in the periodic table they are. Interestingly, those alpha ray emitting particles, the nuclide, radionuclide, has similar mega electron volt. We don't need to care. Every alpha particle looks the same, and similar the half-lives, 10 days, 11 days, and 7 days. We don't need to care. So in case of beta, it's really, and astatine is said to have a higher linear energy transfer. So then we need to understand the linear energy transfer. This one. This is from some very good radiation physicist. I mean the radiation biologist or physicist. He gave a lecture and he let his slides available in IAEA site. Yeah. Unlike beta ray, similar. Why? 47 nanometer, micrometer, and 85 micrometer. Does it have any difference? That kind of difference is kind of not a big deal. Okay, no big deal. And then LET is 80 kilo electron volt per micrometer. 61, no. Unlike beta ray, the alpha ray is so simple. We don't need to, I think, I guess, I estimate, I guess, I predict, I speculate. There is no need to. Finally, our physicist colleagues, they are saying that please don't try to memorize those differences, and all are the same. Okay. And here, astatine is not 211. 211 is only commercially available, you know. But here is a 217. This is kind of awkward information. Francium, we don't need that. Bismuth, we don't need that. What you need is just kind of actinium and astatine and radium. And here, and why the literatures are giving us the different information is because of this decay scheme. 58% is doing the electron capture, and then here alpha particle 100%, and 42% is directly alpha particle, and those alpha particles have a propensity of 42%, and then 58% is here. So the astatine is emitting 5.8 mega electron volt, 42%. One half is 5.8. Another half is 7.4. So that's the reason why we have energy 6.7. Okay, that's average. Tissue range is micrometer. So it's not just several millimeter and then several centimeter, not like that. So now I'm going to take a break. Why? Because you have been given, already you would have been overwhelmed. So I think we need to just take a rest so that I can just kind of, wow, this kind of information, should I memorize all these, or should I just kind of take it mine, or I don't want to enter this field. Why? Because this seems to be too complicated. Once everything is solved and everything is on the commercial and routine base, then I would just kind of adopt this discipline. But if you just choose the later one, you need to just kind of become to child again, and grow again, enter medical school again, and to take nuclear medicine again. So I guess there is no way to just kind of ignore, okay, the current trends. So why not just kind of take save some time to let you understand on your own, and then how much so that you can assure you understand which radioisotope to choose for the best benefit advantage of your patient. That's our responsibility. Interestingly, in this field, study is our responsibility. Study and understanding is our responsibility. We don't, we cannot copy the already established knowledge. So there are much to understand. Okay, I will come back, and then the effectiveness is just one slide, and dosimetry is one slide. Safety is just several slides, and then your direction is you can just get relaxed. Why? Because it belongs to a special place like Korea, China, and Singapore, where nanotechnologies has prospered. So it is not your fault if you cannot follow, you cannot have to achieve the radionanomedicine. So it's not your fault. And however, in case of the molecular therapy, if you just kind of give up, and then it's your or our fault. Okay, let me come back 10 minutes later. And 1:35. Okay, take some coffee or just to take some rest. Okay, welcome for coming back. And during the first hour and a half, I explained interestingly 20 slides, and we have 28 slides, but the later half would take less time, and then I'm just checking. Okay, so and then I have found nothing for chatting, meaning no question now. Now, so maybe you would have been overwhelmed during your medical school years. You would have learned the radiation biology from the radiation oncology, and in my case, an first year graduate student, and we learned from a physicist, and he tried very hard to teach us the radiation biology. And after several decades, I think he does not know anything about radiation biology. He's just a kind of a radiation physics physicist. So the same thing would have happened in your the medical school years. So meaning that today this might be your first lessons, first the education regarding this. It's okay. You know, I have devised my own explanation to teach myself. Why? Because 30 to 40 years ago, radiation oncology was in its infancy, and we did not have any radiation biologist at the time. And that's the reason why I chose, after choosing internal medicine and endocrinology, and then nuclear medicine. Why? Because nuclear medicine, there is many things to be done. So I hope you would have chosen nuclear medicine because it is not well established, but because not because it is well established, but because it is not well established. Okay, so and then I am going to move on, and the second part, you might just kind of lean backward and then just to listen. Do not bother your brain. Do not just kind of harass your brain. Just to listen, and then you're going to you seem to learn more for the later half of my the presentation. And I'm saying it's a presentation, so you might just kind of send any question via chatting. So if I think I am able to answer some questions, then I would just say. If not, then I would confess that I don't know this thing, and then I will study. I'll consult, or if I find myself to understand what you meant, and if I think I'm entitled to give any answer, then today no, then later on, next semester or the next to the next semester, I can just they give you. And then effectiveness evaluation, new therapeutics. Nobody is saying that effectiveness evaluation should be done. No, many things have been proposed in the literature. So I just found only one. In human survival, overall survival, cancer-specific survival, which means the progression-free survival. This we are very much accustomed to. And then I learned the quality of life when I just was studying the nuclear cardiology. But the same quality of life is used for tumor. And surrogate endpoint, which is quite familiar to us, then resist and persist. And then what about the experimental animals? The same survival, but in experimental animal, there are term tumor control probability, as I just told you 1 hour ago, and tumor growth delay. And tumor growth delays was totally new to me just 10 years ago. This is this. And tumor control probability is like that. Tumor control and tumor growth delay is a little bit weird. Tumor growth is inevitable. However, after some radiation, growth has been delayed. This is kind of progression free or something. And here again, natural log survival fraction, telomerase delay, the multiplied by beta y, I don't know. That's it. Then what about the dosimetry? We need to do individual dosimetry. Why? It's because, let me just do, sorry, to the mute everyone. Oh, at first mute everyone. Yeah. 마이크 지금 꺼졌어요? 여보세요? 김동빈 씨, 김동빈 씨.
네네. 방금 말씀 중에 마이크가 음소거가 돼 있어가지고. 네.
아니, 그러니까 저 두 번째 시작한 다음에 계속 꺼져 있었어요.
아니요. 방금 전에 꺼졌어요.
예. 그게 이제 보니까 저절로 꺼졌네. Okay. So I found that there is a gap. So individual dosimetry is needed for newly developed therapy or theranostics. So for individual and thyroid cancer, we don't need individual dosimetry. I'm going to comment on that. For individual dosimetry, at first we need to understand the distribution of radiation and their temporal course. Okay, and for understanding the dose from those distribution, we need a simulation, which is called Monte Carlo simulation. For one case, 200 days are needed. Professor Jae-Sung Lee has made it 3 to 4 minutes. Last year in Jeju Island Theranostic World Congress during Dr. Konienberg from the Netherlands, he was so surprised. He was so envious of Professor Dr. Jae-Sung Lee. And Dr. Jae-Sung Lee said that these technologies can be really open to everyone. So Dr. Konienberg seemed to be relieved that this technology is not going to be protected by the intellectual properties. No, this even though this is an intellectual property based on deep learning studies, it should be widely available. So I'm going to twist arms of Jae-Sung Lee, even though he himself is going to be willing to just to share. If anyone wants to use that kind of technology. And then safety and new direction. Safety, so normal organs should be protected. Kidney, external beam 20 gray, 2 gray fraction is the maximum to the kidney area. But in case of yttrium DOTA octreotide, or the threshold is a little bit higher. It's kind of empirical one. The bone marrow is said to be 2 gray. This is derived from Dr. Konyenberg. Tumor, it's just pancreatic NET 50 gray adjuvant. NET median to achieve volume reduction is 232 gray. So liver metastasis 167 plus minus 139. This is ridiculous, meaning we are not doing best. So here, bone marrow and kidneys and renal dosimetry is using phantom, and repeated imaging can damage even indium or progress-free survival and tumor dosimetry, and that kind of issues are from Michigan, from Netherlands, and from United States, Johns Hopkins, and from Sandstrom, from Sweden, and that kind of is the current situation. And prediction of the actual observed dose is based, should be based upon physiology. Why? We need imaging, and the next the field or discipline of therapy should be called Theranostics. Why? Because we need to predict the response to the benefit and advantage of the patients that we care, whom we care. Okay, so equivalent to uniform dose, BD, I explained. You now have the every jargon, every terminology to refer to. So please don't be intimidated. Let me just kind of do the mute again. And let me just 공유 중지. Stop the sharing, and then my audio is automatically gone. Okay, then for Theranostics here, there are several INT, imaging and therapy, by the Hans Wilgenbester group, or Uwe Haberkorn group, German Cancer Center, or the Technical University of Munich, either. And I'm going to advertise the N-GOL and D-GOL. As I just told you, glutamate urea lysine is linked with the NOTA. Then N-GOL is labeled with a gallium 68, and D-GOL is a DOTA. DOTA-GOL, for some reason, an electrophilicity or something. The chemist colleagues, they educated us that the D-GOL is for lutetium 177, and my eminent chemist colleague in a company and also at a private university, he told me that actinium-225 and F18 compound, for with the same molecule, once it is labeled F18, then it is going to be used for dosimetry or the prediction, and actinium-125 is going to be why alpha emitter is not associated with the any gamma ray from actinium. So unlike lutetium. So that's several examples. And NGUL and DGUL is on clinical trial in India. Why? Because in India, the 1007 or 617 compounds developed from in Germany is already in use. So they comparing those two, GUL and GUL. And so this is my opinion. That's kind of my bias, and you can just adopt it or not. In 2016, any new therapeutics, the mostly monoclonal antibody or tyrosine kinase inhibitors, any new therapeutics, if it is going to be submitted for approval to United States, US FDA, they have announced that new therapeutics should be submitted for approval for with in vitro companion diagnostics. And there has been a release of a document for the industry. So you can just find it. So I have a very good colleague in the College of Pharmacy. Is one of the major the proponent of the in vitro companion diagnostics while he is developing the monoclonal antibody for cancer therapy. And we just kind of reached the conclusion that nuclear medicine is in vivo companion diagnostics. Once therapy is established, then the in vivo imaging is associated with that, so that to do the dosimetry or to do the prediction. And I have summarized this idea in Nuclear Medicine Molecular Imaging with Professor Chan 1 year ago, around the time of the TWC. And this is what I'm saying, thyroid cancer. Even though you are quite an expert in thyroid cancer, your thyroid cancer, the expert, not nuclear medicine. Why? Because thyroid cancer mortality is always the same since in my country 30 years ago. There has been because of the introduction of ultrasonography, the national the health screening for thyroid gland, incidence has increased. Incidence was just kind of ultrasonography related one, not just because of any kind of external one. So this was published a while ago in New England Journal, and we came to be a subject of ridicule or disrespect. Korea is wasting medical resources to so the kind of issues. And then there has been a paper report. Everybody is referring to this paper, 2017, New England Journal, lutetium dotatate trial. That's the kind of beginning of Lutathera, and so. However, some of the patients are still dying after a long while. It's kind of a torture by hope. We don't know which patient is going to improve or which patient is dying. So we need to say that this is an evidence for prolonged survival in some, and we don't know. And this is not a success. Our oncology colleagues for the last 30 years, they have started just kind of a really toxic chemotherapy. When I was a resident, it was so miserable to see and take care of the patients receiving chemotherapy. And after a while, when I was away to do the nuclear medicine, when I just kind of behold, everything has been changed. Some of the cancers had been already cured. And however, still they lose patients once recurrent after 10 years, or once recurrent after some period using tyrosine kinase inhibitors. Why? Because the cancer tissues are so clever to find the alternative pathway to activate to prolong the survival of the cancer. So there's a race. So the lutein dotatate is just a start. So that's the reason why we are interested in actinium and astatine, or bismuth, or other the alpha emitting. And we had a good time of progress here. From 2004 to 2013, nuclear medicine has grown a lot. Even the national insurance system was allotted to look over and supervise or they scrutinize the reason why. And they wanted to understand whether the nuclear medicine, using nuclear medicine, the doctors are maximizing the profit from the poor patients. So our government has scrutinized around that time, and they published a guideline. Before that, the FDG PET guideline was most lenient among all guidelines all over the world, even better than the US ones. After the introduction of the new guideline, by just adopting Japanese, US, Australian, UK, German, and then find out the joint this subset, and then they have shrunken. They try to shrunk just to 20%, but interestingly, it had become the half, and it did not recover. So we thought this year during the annual Congress, someone has said that we put all the eggs in one basket. So meaning we did FDG PET. We make FDG PET as a prodigy. So if we have developed gallium dotatate or the FAPI imaging or fluorodopa imaging, choline imaging, and the kind of diversification would have made nuclear medicine more sustainable, but we did not. We could not. Why? Because we have only 200 people, and we have only several dozens of chemists and several dozens of physicists in this country. So there was the kind of our shortcoming, and we could just kind of we need to read every day the FDG PET, even 30 cases per physician in private hospital, and they have earned a lot. But after a while, nuclear medicine has grown just kind of for 14 years, 2.5 times. The entire the medical expenses increased 4 times. So we lag behind. So no nuclear medicine residents are seen these days, right? So please do not follow this track. Korea had been the best example and now the worst example, right? So to avoid this kind of a catastrophe, I think we need to just kind of at first here, we need to promote nuclear medicine in developing country, especially low and middle income country, using affordable radiopharmaceutical, especially for treatment. You don't need to do the FDG tests so many. The US colleagues, they are doing 20 million cases per year, 10 million nuclear the myocardial SPECT, and 10 million the other nuclear medicine. And they don't do the brain SPECT or brain PET. They do only oncology studies, and they are best expert in both radiology and nuclear medicine. They even several years ago thought about the ending the residency program for nuclear medicine. We were so surprised, and I myself have sent them you have built Rome, city of Rome, or no, empire of Rome, and then you are trying to just to demolish, destroy the Rome at one blow. And then the legally approved specialty or discipline of nuclear medicine in a country like South Korea is going to follow you. And then they have changed their mind. So for the follow-up countries like the LMIC, low and middle income countries, to become the upper middle income or the high income countries, please don't do that. From the beginning, do not lean on correlative imaging too much. Right. Okay. So what is theranostics? My definition is a little bit different. If therapy exists, nuclear procedure can guide therapy. If not, nuclear theranostics shall lead to discover of novel therapy. And it includes brain and heart disease and tumors. Brain and heart is going to be treated by nuclear medicine procedures. No, we have cytotoxic radionuclide, which is not compatible with the brain and heart disease. However, we have very good one. In case of existing therapy of brain, here, epilepsy. Yes, there is a very good surgery. The people are saved. And we do when MRI is normal, we do PET. And here. And then the cerebral vascular disease, which is so prevalent. And for Asian too, if you increase the life expectancy, and then the the kind of aging society, aged and super aged society, if your economy is going to be the responding to your wishes, then you are going to experience the same as ours. And then carotid artery stenosis is going to be more common. And there needs a bypass surgery here. Sorry. Bypass surgery for these patients. And then we can evaluate that here. left posterior the cerebral artery and left the middle cerebral artery territory is suffering from stress-induced ischemia, vascular reserve problem, after successful bypass operations. So that's the theranostics. This kind of diagnostics is good. In that sense, FDG PET for tumors are good, okay? But FDG PET, if you do too much and rapid growth, the 30%, more than 30% every year, then it is going to be scrutinized according to the rules and regulations. And then the insurance company, national insurance system is annoying for every case in every clinicians, and then the clinicians are so much bothered, and they stop to do that. They need to do the MRI or CT, but if they do the PET, then they are going to explain why, based on the evidence-based medicine. So they'll say, the case in my country. In case of existing therapy of heart, here, this is correlative imaging. Okay? So correlative imaging only includes the head and neck to just a lymphoma? No, it's only for a tumor. Every nuclear medicine physician, except for US ones, in USA, nuclear cardiology is done by cardiologists. The US trained nuclear medicine physicians, they don't know anything about the nuclear cardiology. And I found that when I visited the several places, including Indonesia, the current, the one is thyroid therapy and bone scan and myocardial SPECT is and FDG PET is in the verge of introduction. Okay, so stent was inserted. Stent treatment is already there, so why not do the myocardial SPECT? And here, novel therapy of tumor is the kind of narrow. It fits in to the narrow the definition of theranostics. Okay, and then previously these images were taken after sending patients to Malaysia and then coming back to my country, the DOTA tape, rotation DOTA tape. However, these days we are doing DOTA TOP. And cool studies are new direction. One of the new directions, radionanomedicine. This is the conclusion slide, and I can stop now. But if I stop now, then it's kind of too much simple. So I have included 10 more slides here. New direction, radionanomedicine. That's combined nuclear medicine and nanomedicine. So for theranostics, feasible tracer kinetic. So why? Because nanomaterials was labeled with radionuclide, and we do the usual PET and SPECT. And because we are using nuclear medicine, no pharmacologic amount of nanomaterials are going to be administered. Trace amount and trace kinetic is the core theme of nuclear medicine, and which has been rendered to nanomedicine. And 10 years ago, our nanomedicine colleagues, I'm in charge of the vice president of International Society of Nanomedicine, which was incubated by the European colleagues, mainly Swiss colleagues. And 10 years ago, when I first admonished, when I just advertised or publicized this radio nanomedicine, they were puzzled. And does it belong to the just nuclear medicine? Yes, it belongs to nuclear medicine. We are doing, even Hans Wilhelm Wester, the best the radiopharmaceutical scientist in Germany, one of the best, and he is doing this small molecule. And also Jaemin Jung is also doing this small molecule. Somebody has been doing the albumin, we too, and WBC scan, and that kind of issues. But what about the nanoparticles? It's just a small molecule has a, even though we have a library, but it's limited. Then that library chemicals can be loaded here, antibody or small molecule and whatever. Recently, albumin was labeled with a mannose, or albumin labeled with the galactose. This went into only the copper cell. Galactose is hepatocyte. The kind of selectivity was so marvelous. Even though the particle is there, mannose is just kind of targeting copper cells and sinus endothelial cells, liver sinus. So that's so actually this is nuclear medicine. We have just expanded from small molecule to nano, and Jaemin Jung has used even though he knows that the quantum dot cannot be used for a human. Why? Because it's made of the toxic the molecule atoms, such as yeah, I forgot many toxic. Does anyone know that how much amount of gadolinium is being given to the human? There is none. If I calculate, if I remember correctly, they are giving 6 grams. We are giving 40 microgram. Here, 1 milligram is divided 5. 1 vial is 1 milligram, microgram. So huge amount. That's the reason why the gadolinium is causing nephrogenic systemic sclerosis. They are using pharmacology amount. We are using 0.05 picomole. And then the cold compound was given here. You are seeing the same gallbladder. Okay. I told you mannose and galactose. Once here, not this one, here, that kind of sugar moiety is giving the specificity. This is an unpublished data, okay, unpublished data. And copper 64 and fluorescent dye, co-labeled ones, were given intravenously, PET imaging first. One is showing the gallbladder, the other is liver and spleen. And then after sacrificing the animal, ex vivo fluorescence microscopy is showing this kind of really marvelous differentiation of here. Here, the copper cell, here is the sinusoidal. And then hepatocyte is being burdened by Dr. Edison is coming out and in several times. Okay, I now remember your name. And what kind of nanomaterial? There are so many nanomaterials, and if you successfully engineer nanomaterials, and then this kind of best journals, Chemical Society, or the Advanced Functional Material, mostly impact factor is 13 or something. They are publishing your the materials, and so many. How many? With hype. They are saying that their nanomaterial can be used for any kind of biomedical imaging and therapy, photodynamic therapy or whatever, and which is not accompanied by any experiment to prove that. So previously they have used the gold nanoparticle, and these days we've used the iron oxide nanoparticle, and that's the subject of Professor Soo's PhD thesis. And Professor Soo, Dr. Soo also helped me to publish. He read every manuscript from the first to the. He read every references, and finally he got his PhD using this concept. Here he labeled the iron oxide nanoparticle copper 64, and then found out a very interesting one, which is still yet to be published. So, during warmth, he explained why, because his thesis was passed last August. So his thesis is online. So we explained for December the warmth meeting, which is going to be virtual and presided by Ryan Hussain, Professor Ryan Hussain of Bangladesh, and we explained, and we already recorded. So we don't know when. Maybe the first week of first or second week of December. Wow. And what might be the kind of physiology? Physiology meaning not gram, but microgram. So there is a patient, which was very important. Sorry. A little bit wait. Wow. Let me just undo. I need to just stop. Okay. All right. So because we all of us are the physicians, we don't bother bother about the noise which is caused by the patients. Okay, so that's the reason why we are doctors. Okay, so physiologic ones, and not gram, but a very small amount. And in mouse experiment, a very small volume, very small, the compatible, the protein concentration. Why? If not, CSF physiology is not going to be represented in our experiment. Okay, so Dr. Asmar, who has spent 1 year and 2 months here now, this is his product. CSF is to cervical lymph node. This is superficial cervical lymph node, and these are deep cervical lymph node. This is a new concept which was based upon 2015. After 100 years of debate, finally the relevant scientists found and published the finding of lymphatics in
혹시
brain.
교수님 혹시 화면 공유는
안 했네.
아, 네네네.
할게요.
아, 네, 감사합니다.
네. 화면 공유 어디서부터 안 했나? 기억 안 나네.
한 15초 전에 네. 네.
아, 그러니까 그 어떤 슬라이드부터인지 좀 보고 있었으면은 얘기해.
네네네네.
아, 이게 이 환자가 시끄럽게 소리 내는 거는 어쩔 수가 없다고 그 얘기하느라고 까먹었어요. 공유. 안 보고 있으면은 누가 일깨워주기 전까지는 모르겠네. So all of you have seen this Dr. Soo's story, success story, and then finally here I have one or two more slides, so you can get relaxed. So as I promised, the second part was a little bit more about the science fiction type of story, or National Geographic or Discovery Science type of. Here, Dr. Asmal did this experiment and while understanding physiology. Physiology is a best policy of nuclear medicine. And 2015, after 100 years of deep debate, finally they found that the lymphatics are there in brain. So so far the CSF is disposed, cleared from arachnoid villi within the superior sagittal sinus vein of the meninges of the brain is wrong. And actually, this is not wrong. They think that the 10% are still cleared here, but what is wrong is there is an lymphatic here. That lymphatic is just kind of receiving CSF, extra CSF, or tau protein and whatever from the interstitium to CSF to lymphatics, and finally lymph node, and then thoracic duct to systemic circulation. But this is the mouse or rodent case. In humans, they do not use the nasal mucosa. There was a recent report in scientific reports, in the Journal of Scientific Reports. And we can do the quantification and imaging. And we are going to evaluate everything based on this, especially for Alzheimer's disease. So far, Alzheimer's disease imaging of tau and amyloid is available. This is the answer to the question. In Hassan Sadikin Hospital, several promising and also glistening and also vibrant and also really being who would be in charge of the next generation, the young faculty members surrounded me after my just kind of a visit. In a corner of the auditorium, the classroom at that time, one of you asked me, what's the prospect of the nuclear neurology? And should we follow the Christopher Rowe or other the pioneers, tau or the amyloid imaging job? Should we just kind of find out the way how to follow them? This is my answer. Tau and amyloid imaging is doomed. Why? Because tau still the unidentified for its kind of efficacy. But in case of amyloid imaging is too prevalent, even in normal. And once amyloid was treated in many clinical trials, including the most recent the Biogen's aducanumab trial, contradictory results were there. That's this slide was made 4 weeks ago. 10 days ago, in Bloomberg News, just near CNN, Bloomberg News has said that the Biogen's aducanumab failed in FDA review. So the contradictory results in brain swelling, FDA currently, according to the current information, rejected. Then amyloid, the Alzheimer's disease does not have the treatment. So if there is no treatment, we need to just give the treatment, or we need to just give the method to evaluate the new treatment. So if new treatment is waste disposal, better waste disposal, then we need to just kind of, that's our idea. Okay, ISCSF, perilymphatic space or whatever. And this was the reported to clinical nanomedicine meeting 3 weeks ago, virtual online. And then the parsimonious explanation of pathogenesis. Pathogenesis is changing. The amyloid and tau. Amyloid is triggering tau. Among these two, we choose one. I would choose this one. Based on the literature search, A beta triggers, and then is going to become going to be disposed to the trash, which is called amyloid plaque. Okay, extracellular tau. Tau is an intracellular microtubule-associated peptide protein, but it is frequently mutated and truncated and phosphorylated, and sometimes it is excreted in extracellular vesicles and then from microglia and neuron and astrocytes. And the 2020 Nature Neuroscience and microglia, role of microglia is emphasized here. And within exosomes and the tunneling nanotube has the similar constituent. And then that nanotubes is used for cell communication, and also exosomes are too. And extracellular taus are excreted, naked form, or exosome, ectosome, and tunneling nanotubes. So our goal is a tracing of tau. And then we are going to use the albumin as a surrogate. And then this, you have already seen this one. Why? Because 7 years ago, and published 55 years ago for the first time in the world, just like WBC, the HNPO WBC imaging, we successfully labeled and using technetium HNPO, the exosome, and then find out their distribution. This technology is going to be used. And this might be my last slide. Technetium is easy, but people can say that once given to the in vivo, and exosomes are going to be punctured and deteriorated, and then technetium, hydrophilic technetium HMPO is going to be leaked out. Then what we are seeing might be the hydrophilic, the released technetium HMPO or not. We discussed in the discussion that, so we made another method, copper 64 labeled one, and also surface was modified with a collaboration with the Harvard when Professor Huang was there. And also we just injected to the striatum here. We found that it's not going anywhere. It is there. So I don't think the exosomes are working as an endocrine. I think the exosome is the paracrine mediator. So, so you have listened to the kind of from the textbook type of knowledge, which is not listed in the textbook, and you can just refer to my interpretation, and you can correct it. If it is wrong, I would be happy to find my the wrong understanding. So if somebody is explaining, or somebody is just indicating the incorrect part, immediately I would change my mind and understanding. Okay. You are in charge of future. Okay. There's no way. Okay. So now I have yeah. So according to the precedence of Professor Edmund Kim, I have 25 minutes of discussion. However, I'm going to use only 10 minutes to make you kind of a little bit relaxed. So let me find out if, yeah, we have chatting. Yeah, we can see my screen. Oh, I solved it. Okay. Kim, our manager. No questions. Wow, Andreas Lim. Yeah, good morning. Yes, good day. Thank you. And Dr. Gil, you are requested to ask any question. It's kind of a fixed discussion. And then Dr. Asmar should follow, and then everyone. Why? Because Dr. Gil and Dr. Asmar is in my class. Dr. Gil, everything is clear. I found that you are already well knowledgeable about the nuclear cardiology, but I'm curious if you are already knowledgeable, you have many knowledge about the molecular theranostics too. No? You need to unmute.
Yeah, actually, I don't know well about radiation biology that you, but I think I could manage it myself, so I can
Yeah,
get the question. Yeah.
Yeah, okay. So you mean that you can just kind of, based on my the materials and my introductory the presentation, you can maybe in the next semester, you might just kind of challenge, or you might improve the interpretation or understanding. And anyway, you can use this kind of information for during the our morning conference, and I found that the knowledge regarding molecular theranostics or the therapy clinical trials are scattered and fragmented between faculty. Professor Chun does not know everything. Professor Zhong does not know everything. Professor Peng does not know everything. Dr. Bae does not know everything. Professor Seo does not know everything. Professor Choi Hong-yoon is just kind of would have escaped from the discussion. Why? Because he used to be very excellent, but in this subject is too much for him in his time. So meaning that you are the one, first year resident, you are the one to just kind of integrate all those information and all those clinical trial results, and or the Selbion's N-goal and D-goal stuff, or future chems are the one approach, Professor Chi, Dr. Chi. So you are the one. If you just excuse yourself from the learning that kind of issues, then we don't have any future. Okay. So, okay. So I'm really happy that. Then Dr. Asmal, do you have any comment?
Sir,
Oh, yeah. Okay, Dr. Asmal. So in the same room, the only one person can just the unmute.
So the about the yttrium-90 and lutetium, is it because because the lutetium's energy and range is less, so it can cause less damage to normal tissue surrounding the tumor?
Aha, yes, yeah. So let me just kind of very good point. Yeah. So while I just trying to answer the where is it? Answer to Dr. Asmar's the comment or the question. Please formulate your question. Meaning the first or this one, not this one. Where is it? Yeah, this one. So Dr. Asmal is already reaching the conclusion to be submitted as an answer to the graded course term examination. So why rotation? He pointed that not just thinking about tumor. Tumor, this might be good, better, but what about the normal tissues? Tissues like kidney. Lutetium, the peptide compounds are excreted to the kidney. Kidney is a critical organ, right? So his point is this. And then my point is I have a before this presentation I did not went on to that kind of no, not this one. Maybe Dr. Gill, you need to just understand this, and Dr. Gill and Dr. Asmal, at least two of you need to read this. Okay, and from their data, I just put this kind of calculation. This is my calculation. They have just wrote down only the proliferative rate constant and then lambda effective. But what I'm trying to add to you is that here, lambda effective over lambda effective plus mu repair. How to look at this equation is, it's like this, 1 plus effective. So even though I put the like this, I have insinuated that lambda effective, this is a single parameter, and if this is high or if this is 0, it's that kind of issue. So even though it looks like the complex and even this kind of actually this is, and actually this is not, right? This is what we are, and this looks complex, but actually it's lambda effective mu. The rate constant ratio is important. +1 is just kind of a dummy. And that ratio is important, and meaning mu rep and lambda effective of normal organ and tumor might be different. Okay, and mu rep repair constant is sure to be different, normal and tumor. That's the essence of how to start your reasoning. Dr. Asmal pointed this out, intentionally or without the implicitly or explicitly. I made Dr. Asmal's comment or question explicit. Dr. Gill, have you understood what was commented by Dr. Asmal? Correct?
Yes, yes, Professor.
Yeah. So that's the point that I did not just comment. And after the first half of the presentation, during the 10 minutes of break, this point, missing this point was pop up in my mind. If I just add this, then the next generation, the promising next generation young physicians should familiarize this concept more and better. Okay, next week you are going to, all right, so is there anyone to have any comment? Oh, I have silenced everyone. During the Professor Edmund Kim's lecture, there have been so many interactive discussions. Everything is so clear. No, everything is so obscure. I'm so sorry.
I'm so sorry.
If I have a concern, don't worry. Itu anu tuh. Itu apa anu? Nanya. Oh.
Okay. Okay. Let's finish this one, and you can just anyway. Next week, Professor Ha is a young the faculty in St. Mary Hospital, the Catholic University. He has become the professor last year, and he's going to just explain how we get to know the concept of the brain image analysis, and which is so. Even though I just kind of tried to made believe, and I pretended to just to reprimand the blame the FDG PET for tumor. So it's not actually that's 80% of our life survival, but we need to just to expand. We don't just kind of get a standstill. All right, so these days FDG PET CT is so clear, and then interestingly, policymakers who had been involved in the guideline making guideline. Once they got ill, they want have a PET CT. so it's kind of a ridiculous situation. Okay, see you. Actually, see you next to February. Okay.
Yeah.